I got him. Thank you so much. I appreciate you guys. All righty. So it's gonna get faster now because we went over the bulk of everything. Okay. So who could remind us what are cell adhesion? These are the staples. You know what makes your tissue histology when you look in a nerve tissue, all the neurons are tightly packed, all the muscle cells are tightly packed, all the epithelial cells are tightly packed. Well, they don't become tightly packed on their own. They are stapled, hooked with adhesion. So there are different adhesion molecules, caps and cams, caps and cams. Cap is cell adhesion protein. Cam is cell adhesion molecule. Caps and cams. So who can tell me what is the function of the cell adhesion molecules? Cell adhesion molecules. So they stick together what? We can stick together what is this? So if you look, integrin is like a hook. You see how, like a nail, it's hooking the epithelial tissue to the basement membrane. You see how we hook in the endothelial cells to the tunica intima and the tunica media and the tunica externa. So things don't happen on their own. We need to have these cell adhesion molecules. And we have different types of molecules that are responsible for the adhesion. Sometimes we have cell junctions. Cell junctions, there are three types of cell junctions: tight junctions, desmosomes, and gap junctions. Gap junctions are my favorite because not only are they hooks, but they're hollow from in the middle. So why would you need something like that? Why would you need two cells hooked together with a nail, but the nail is hollow from the middle? So that can what? Yeah, so the passage of electrolyte, the passage of glucose, nutrient, waste. They need to communicate these cells. Cells are not like how homes are here. You have your front yard, backyard, your fence, then your neighbors. No, homes are like back home in our countries. The houses are really tight. They even share a wall. And sometimes there's even a door. You can go from your home to the home of your neighbor. Your cells are just like that. They need to communicate with each other, especially like nerve tissue. I mean, that's a type of a tissue that needs to be in unison. So when the action potential arrives, all the cells get depolarized. If you don't have a gap junction, how is the sodium going to follow through? How is all of the electricity going to be passed by? So that's a gap junction. Sometimes you don't need a gap junction. You just need an actual junction that is not hollow from the inside. This gating enables the uninjured cells to protect themselves from injured neighbors. So you see, these are the hooks. You see? Cellular communication. Remember, that's one of the signs of life. Cells need to communicate, to talk to each other. Like, for example, especially in the immunology. In immunology, your white blood cells, they will take a fragment of the bacteria and they would present it. Remember the antigen presenting cell, APC? What's the antigen presenting cell? It's a cell that grabbed the fragment of the bacteria and presented it to those really big cells, the B lymphocytes and the T cells, and then they will recognize it. So they need to communicate cell signaling by contact, sometimes remotely, by sending a hormone or a messenger that travels even through the bloodstream. Sometimes it doesn't need to go that far, just pass it to nearby, like a golf. Like just hit it it's gonna buy a receptor next to the cell. Sometimes the communication is through gap. We talked about that, the gap junctions, the hollow cells. Okay. That signal transduction. That's what I said earlier. Remember that. Epinephrine cannot get in. It binds on the outside. Then there is an intracellular signal transduction pathway where there's a cascade involving a second messenger. Like I said, the Uber driver cannot get in the school. The public safety is the second messenger who's going to go. But I mentioned One messenger, it doesn't need a second messenger. You can go all the way from the outside all the way to room 400 right here. What's an example of a hormone that can go without a second messenger? You can go all the way to the nucleus from the outside, penetrate the plasma membrane, swim through the cytoplasm, enter the nucleus. What's an example of that? Testosterone and estrogen and aldosterone. All of these, why it can do that? Because it is made of cholesterol. It has aldosterone, estrogen, testosterone, because they're all made of cholesterol. And you know, when you're made of cholesterol, you're a lipid, you're oily. Who else is lipid and oily? Your plasma membrane. So they dissolve in each other. Testosterone will smoothly swim through your plasma membrane like no problem, because both birds of the same feather flock together. The same, if you are like, you dissolve like. You're nonpolar, you dissolve nonpolar. What's the name of the hormone that will never be able to cross the plasma membrane? It has to bind on the outside, and it needs a second messenger in a signal transduction cascade. What's the name of it? Epinephrine. Very good. In your fight or flight. I mean, that's the adrenaline, the one that gives you the rush, makes you tense, because you know, if you want to survive, you have to be tense. Heart rate go up, blood pressure go up, contractility goes up, and you need to run for your life for survival. That's why we call it the survival hormone, adrenaline or norepinephrine. Metabolism, we talked about. Okay, so anabolism, catabolism. Remember I mentioned earlier, somebody running on a marathon, and I'm in my room on Saturday eating pancake. Absolutely, our metabolism is not going to be the same. The metabolism for somebody who's running the marathon, they need to burn fuel. So they need to break macromolecules into their little respective blocks and get energy out of that. But me, I'm not burning anything. So I'm storing. That's why I become fat, because you keep storing and storing. But people who become thin is because they're burning and burning and burning. So breaking down, so we have two types of metabolism. We have metabolism, which is divided into catabolism and anabolism. Catabolism is when you break down from really big to small. You have big macromolecule, carbohydrate, you break it down to small macromolecule, monosaccharide. Big protein, you break it down into little amino acids. Big triacylglycerol, you break it down into little tiny glycerol and free fatty acids. And you have big DNA, you have big nucleic acid, you break it down into nucleotides, from big to small. But anabolism is the opposite. Anabolism is from small to big. It's from small to big. So in catabolism, we are breaking down, while in anabolism, we are building. We are building up. We are building up little monosaccharide, little fructose and glucose, build them together, get them maltose. Glucose and glucose, bring them together, you're going to get maltose. Fructose and glucose, build them together, you get sucrose. Galactose and glucose, bring them together, you get lactose. So sometimes your body needs to have the ability to do both based on what is happening. Are you burning? So you break down. Are you storing? You build up. So that's metabolism. All of these processes, communication, metabolism, all of that is involving enzymes. So in the next lecture, you're going to find out that the disease is because the enzyme, the gene is not working. So there is a protein responsible for hooking. Remember dystrophin? So the cells are not going to be hooked so tightly. So the patient is going to have really weak connective tissue. Why? Because is something wrong with the ribosome? No. Something wrong with the translation? No. Something wrong with the transcription? No. Something wrong with the gene we're trying to transcribe. The code is not working. Okay, so ATP this is just to show us that we need the energy. And there are a lot of diseases where metabolism cannot successfully happen. Anybody heard of phenylketonuria, PKU? Yeah. So phenylalanine ketonuria happens when the baby is born with a mutation and a genetic defect that we cannot recycle phenylalanine. So when the baby eats food that is rich in phenylalanine, the body now cannot metabolize it. So phenylalanine is going to build up and build up and build up and deposit in the brain, causing intellectual disability. So what's the solution? Don't eat food rich in phenylalanine. It's not that hard. But can you cure it? No, because you cannot make a gene. You cannot make a gene. So the worst type of diseases are the congenital, because those who are not inflammatory, you cannot use anti-inflammatory, antihistamine. You cannot use chemotherapy, radiation. This is something that, you know, God, he gave it to you. You have to deal with it, be patient with it. Okay? Everybody has a congenital disease, by the way. Maybe it is not like very severe, but nobody is perfect. If you do a full examination from head to toe, nobody in this room, including me, including you, is 100%. You gotta have something in your spinal cord, something in your. One of the students told me, Professor, I'm gonna go over this when we do the cardiovascular system. She said that she had a tricuspid aortic valve. That's just an anatomical variation. Every one of us here, the anatomy and physiology you learn, this is how your body is supposed to be. But does that mean it will be that way? Absolutely not. Everybody has definitely some structural or functional congenital disease that they have in their body. Maybe at this point in your life, you found out what it is. Maybe you didn't. Maybe you're going to die without even knowing it, but you absolutely have it. Okay, clear? All righty. So this is just to show you an example. When you break down pyruvate into acetaldehyde and to lactic acid, acetyl-CoA, and ethanol, that's an example of anabolism or catabolism. See, you had big old pyruvate, you broke it down into acetaldehyde, catabolism. Very good. Okay, we talked about that. Diffusion and filtration. Okay. We talked about engulfing and endocytosis. See this bleb? That's a bleb. This bleb here is how you're going to bring in something harmful, and then the lysosome is going to fuse with it, and then the lysosome is going to degrade the content. You see here, the lysosome, that's the army, the police, and the bleb has this pink dangerous material where my mouse, you see where my mouse is? You see, this is dangerous. This is going to kill us. So then you had a lysosome, and that's a phagosome. Phagosome containing the dangerous material. Lysosome containing the hydrolytic fighting enzyme. Then they fuse together. You have phagolysosome. And then the phagolysosome is going to destroy and render it into a really harmless bleb. Into a harmless bleb. Harmless bleb. Meaning that is really not dangerous. You first endocytose material that is harmful, and you endocytose the harmful material, and then you have the lysosome is gonna bind with the phagosome. You see, that's the phagosome containing the dangerous stuff we are afraid of, and that's the lysosome. You have two somes: phagosome, lysosome, phagosome, lysosome. Then they're gonna bind together. You got a combo, phagolysosome, and then you're gonna have your harmless bleb. I have a video on that on YouTube. You can watch it. Maybe like at some point I can make it like a out of class assignment. But let me just show it to you. So if you go on YouTube and you type in my name, yeah. If you go on YouTube and you type my name, Okay, and you go to my channel, you go to my YouTube channel, then and you go to videos, you will find I want to show you the video of the phagolysosome. Is very important. This Zoom recording now, I'm going to post it for you online also if you just wanted to go back and kind of like you wanted to listen to it again. But like I told you, the most important thing is the homework. Don't be like my other student the other time, we're going over the lecture slides, memorizing the lecture slides. You could do that later, but the most important thing is the homework. All these questions cold, you know all of them, and you will see you will do excellent. So let me show you the phagolysosome. Where there it is. Okay. So if you look here, no sound here. Okay, there you go. If you look here, you see that's the plasma membrane. See the plasma membrane, and it has all of the. Remember the markers that we said when you give graft rejection, your immune system is going to screen a surface. It's screening for those. Only the identical twins, they have the same markers on the surface. What is this here? This here is your white blood cell. It's gonna, what is this receptor? That's the microbe. You see the microbe? So we internalize the microbe. So now we have a bubble here containing our microbe. What did we call this? What some is this? Is this the phagosome or the lysosome? That's the phagosome. The phagosome is containing the microbe that is going to hurt us. These little knives, this bubble here containing those knives, enzymes, representing knives, degradative enzymes, metalloproteinases, proteolytic enzymes, acid. What's the name of this some here? Is this a phagosome or a lysosome? That's the lysosome. Now they're going to both combine. They're going to merge together. The lysosome is going to merge with the phagosome. And then what's going to happen? Those enzymes are going to shred and they're going to destroy your microbe, right? What do you name this complex of both of them joining together? What do we call that? Phagolysosome. And then the phagolysosome is going to secrete our harmless blebs. So you can watch that at your own pace. Okay. And you will see me just talk about it again and again and again. You understand? So this is what the outer classwork video summary is going to be like. I'm going to assign one like this. So you're going to sit down at home and just listen to it. And then you just make a summary of like, you're not going to do like AI or no two people going to share the same one because you're going to be detected with turn it in and you're going to get dismissed. So be very careful. You just listen to it. You type your original, authentic, understanding and summary of the video, and then you will see that I will give full credit as long as I see that when I read it, I know what I talked about. You can't go on like Chat GPT. I really know all that stuff. You can't go on Chat GPT and say, phagolysosome, and you look in here, toll-like receptor, you put toll-like receptor. I know what I talked about. So when I read it and you have something I never talked about and you have something that wasn't on the video, it just shows that you never listened to the video. You just wanted, you typed it. So make sure you listen to the video. Okay. So that was as far as the phagolysosome. We talked about electrical impulses mediated through the extra potentials, only going through the heart and the neurons. And when it comes, so now we're moving into the cancer. So the cell cycle, some tissues always enter the cell cycle, like your skin. You scratch your skin, new skin come up. But some tissues will never enter the cell cycle no more, like your brain. If your nerve tissue is regenerative, it can enter the cell cycle. You will never have a stroke because the nerve tissue die, we'll make more nerve tissue. But that will never happen. Why? Because nerve tissue is non-regenerative. Skin tissue is regenerative. Bone tissue, nobody broke their bone and the bone never healed because bone tissue is regenerative. But heart tissue, is it regenerative? Is never regenerative because heart tissue is terminally differentiated, just like the brain. So you have a heart attack and a stroke. But you never have bone stroke or bone attack because that's a regenerative tissue. Something wrong with the bone, we make new bone. Something wrong with the skin, we make new skin. But something wrong with the brain, you may never make new brain. You can't make new neuron. So you have to be very careful with the alcohol drinking and the people doing the boxing, fighting, like hitting in the head. So that part of your brain died is never coming back. No millions of dollars will give you a new brain. No alcohol is not gonna, this acetaldehyde is gonna be metabolized and it's gonna cause oxidative stress in your brain, damaging your brain. And when you get older, you're gonna pay the price because your brain tissue is non-regenerative. The skin tissue is regenerative, okay? The phases are, there's another video in there about the cell cycle. And you need to know the cell cycle because we're going to talk more about that when we do the cancer lecture. And you're going to see that the problem is this cell cycle, you're going to go from one stage to another. There are cell cycle regulators. They first examine, inspect, is the cell ready? Are all the proteins healthy? Is the DNA healthy? We don't want to duplicate a DNA that is broken. We don't want to duplicate a DNA that is mutated. We want to ensure all the DNA is healthy. But when something goes wrong with these proteins due to a mutation, then the cell keeps going awry and keeps giving us new cells because there are proteins like break. They stop the cell, say, hey, cell, enough. We were injured, now we healed. You don't need to make more skin tissue. But when they get an earring and they get the big keloid, have you seen the keloid? That is because a mutation happened, and you see the scar tissue that's forming, and there is continuous mitosis because something is wrong with the protein that's supposed to. These are called tumor suppressors. We will talk about that next lecture. Okay, all righty. So this lecture is just lecture. So on our syllabus today, we have our agenda is 1, 2, cell biology, genes and genetic diseases. So now this second lecture is going to be really fast. The first one, we need to spend a lot of time reminding ourselves what is the cell, the nucleus, the genetic disease, the congenital diseases, and all that kind of stuff. So now we're just going to be going over what the diseases are and making references to what we discussed in the lecture. So your first exam is going to be on the 26th. You're going to have the exam, and after the exam, we're going to have the lecture. So it's going to cover cell biology, genes and genetic diseases, the homework, pretty much the homework. Then the homework for the cardiovascular system. By the way, all are up now. All are up. And lastly, the endocrine system. And then you're going to come 50 out of all of those, 50 questions. Which 50 questions you will never know. Students say, I did it from the top ones, from the middle ones. It doesn't work that way. You have to understand all the questions. And then 2 weeks later, February the 2nd, February the 9th, we're going to have lecture exam 2 on everything. It's going to be 50% on lecture exam 1, which is cell biology, genes, cancer, cardiovascular, endocrine. And the other 50% is everything that has not been tested after, which is the respiratory system. Then lecture exam 3 is going to be, then the midterm is going to be 50% from lecture exam 2 and above, and 50% after lecture exam 2. And so on. We're going to be going like that. So every exam is going to be 50% that wasn't covered and 50% from the latest homework and above. We will talk more about that when we get there. For now, you need to know on the 26th that homework, you got to be like Peralta, and you got to do all your homework and review it and understand all the questions so that on the 26th, when I do the Scantron, I keep seeing 50, 50, 50, 50. There's no reason for you not to be getting 50 out of 50. And that's the first exam, only this. The next exam, everything. And it keeps getting harder. So make sure you go over the questions. Is that clear? Okay. All righty. So now we're going to do the genes and genetic diseases. Yeah. The homework question. What do you mean? for the material, for the exam, there's different things. There's an exam, there's in-class work, there's out-of-class work. In-class work, what we do on the board, and your attendance, and your participation. And out-of-class work, the homeworks, and these summaries that are assigned for you. The exam is the homework. You go over all the homework questions. I'm going to be picking from the homework. You're going to see on the exam, oh, I saw that question on the homework. Oh, professor, I forgot. Well, what are you going to do now? I can't help you. Okay? So I can't take the exam for you. I gave it to you, but I can't take it for you. So you have to study it from before. And you have to really think about it. And when you go do the homework, you will see that it's what we discussed in class. Okay, you need to understand that the human chromosomes, we have two different types of cells. We have somatic cells and we have germline cells. We have two types of cells. We have two types of cells. We have somatic cells and we have sex cells, also known as germ cells. And these are the sperm and the egg. Two types of cells. Somatic is every other cell type that's in your body that is not sex cell. That is not a sperm, that is not an egg. Is that clear? Your muscle cell, is it a somatic or a germline sex cell? Your neuron, is it somatic or a germline? So now each cell, like we discussed earlier, has a nucleotide, has a nucleus. Inside the nucleus of all somatic cells, you have 46 chromosomes. But in the sperm, you have only 23, and in the egg, you have only 23. Why? So that when they combine, you have a zygote that is the sum of both of them, which is 46. This is really, really, really important. You need to know that all the somatic cells are diploid. Diploid, 2x. All the somatic cells are diploid, 2x. While all the germline cells are half, are known as haploid. 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 So they combine to give you the zygote. So now you need to know we have 4 different types of macromolecules. You really need to know that. What are the 4 molecules that exist in life? You're either a protein. What are proteins made up of? Amino acids. Yeah, peptides. So we need micromolecule, macromolecule. Micro, macro. So the micromolecule, amino acid. The macromolecule, protein. That's one of them. What's the second one? Sugars. But you know, we don't say sugar. We say monosaccharides, disaccharide, polysaccharides. Monosaccharide, like glucose is a monosaccharide. Not sucrose. Glucose is a monosaccharide. Fructose is a monosaccharide. And lastly, galactose is a monosaccharide. Then you combine glucose and fructose. What do you get? You get fructose and glucose. You get sucrose. That's a disaccharide. You combine glucose and another glucose. You get maltose. That's the whole grain. That's the oatmeal. And galactose and glucose, you get lactose. That's the sugar of milk. That's disaccharide. Then you go up and you make starch. Now that's a polysaccharide. But in the end, a protein and a sugar are not the same. You're supposed to look at the structure and tell, is that a protein or a sugar? You need to know that. Like if I show you a monkey and an elephant, you're not going to say, I don't know which is which. You need to know, how does a protein look like? How does a sugar look like? So we said the first one was a protein made up of amino acids. And then we talked about sugars. Polysaccharides made up of disaccharide and monosaccharide. Who's number 3? Lipids. They are made up of free fatty acids and glycerol. Glycerol, free fatty acids come up together to give you triacylglycerol. And what's the last one? Nucleic acid. Nucleic acid is very big, like RNA, like DNA. But what is the nucleic acid made up of? Yeah, nucleic acid is made up of nucleotides. The nucleotide is made up of phosphate. But you need micro and macro. So you need to know every macro and who's the micro who makes it. When I tell you what's a polysaccharide is made up of, you say amino acid. Now that's a big problem because polysaccharides are made up of monosaccharides. Proteins are made up of amino acids. Nucleic acid are made up of nucleotides. Lipids are made up of glycerol and free fatty acids. So now we're going to break down the nucleotide itself. It's made up of 3 things: phosphate, nitrogenous base, and a ribose sugar. That's why people who are hypoglycemic, they don't have sugar, they cannot replenish their DNA because sugar is part of your DNA. Phosphate is part of the DNA. That's why the DNA is negative, because phosphate is a negatively charged. Nitrogenous bases, there's pyrimidines and purines. Pyrimidines have a big name, but they have only small ring. Purines have a small name, but have 2 big rings. I used to get confused about that when I was in college. I used to think the bigger one has a longer name. No, the smaller one has a longer name. And the DNA is double helix. The RNA is single helix. There you go. You see, that's the nitrogenous base. This has 1 ring. This has 2 rings. What is that called? Okay. 2 ring. What is that? Purine. 1 ring. What is that? That is pyrimidine. Then right behind, so each nitrogenous base is attached to a pentose. You know the pentagon? Pentose, 5-carbon sugar. Pentose. And there's a phosphate. You see? Phosphate, pentose, nitrogenous base. Phosphate, pentose sugar, nitrogenous base. Phosphate, pentose sugar, nitrogenous base. Phosphate, pentose sugar, nitrogenous base. That's the DNA. Both of them, they bind together because they bind together because of hydrogen bonding. Hydrogen bonding is the temporary bonding, like, classmate. But nitrogenous base and a sugar, that's like sibling. They have a very covalent bond, So the classmate breaks and form. Today, you're in this class with this friend. Next semester, you're not with them anymore. You see them again, you're bind again. You don't see them again, you break apart. But your brother, your sister, you're always with them. At least supposedly. Okay. Consists of 2 steps. Oh, we talked about that. You see things overlap. Protein synthesis, genetic suppression. Now you know what genetic suppression means. Genetic suppression means we go to the gene, we transcribe it to give us the mRNA, we translate it to give us the protein. If I want collagen, collagen is a protein. I need the central dogma of biology. Where do I go in the DNA? Do I go to the hemoglobin gene or do I go to the collagen gene? You go to the collagen gene and that's called genetic suppression. If somebody can't make collagen, it's because what? Because the gene is defected, is broken. It's not that they don't have it. We all have it, but something bad happened to it before they're born, or because the mom was drinking alcohol during the pregnancy. Can the dad drink alcohol during the pregnancy? He shouldn't anyways, but he does. Okay. All righty. So transcription, we talked about that happens in the nucleus. Translation happens in the transcription. Gene splicing, this is really important. So when we transcribe the DNA, and we get the mRNA. Is that whole mRNA we're gonna use it, or we're gonna do some surgery, splice it, fix it up? Yeah, that's called gene splicing. So what is gene splicing? Gene splicing is when you take the mRNA and you chop out what you don't need and you leave behind what you need. What you don't need is called the intron. This is really important. So we have, if you don't write it, nobody will remember. So if you look into the DNA, you will see that we have the DNA and then we transcribe it in order to get mRNA. This mRNA is really long. So it has some parts that need to be chopped out, removed, some parts that are not good. So then we do some gene splicing. We do like a surgery, and it's called post-transcriptional modification. When we remove all the brown stuff we don't need, and now you have all the bad brown stuff that we don't need, and now you have your whole fresh DNA, that mRNA that you need. What you need is called the exon. What you don't need, what you need is called the intron. No, what you need is called the exon. What you need is called the exon. What you don't need is called the intron. Why did they call it that? Because the intron is useless. You don't need it. It stays in the nucleus. In nucleus, intron. But the exon is going to exit the nucleus and go to the ribosome. It's going to exit and go on a cruise. It's going to go on the cruise to the cytoplasm. It's going to go out. So you really need to know the intron and the axon. So from the name you can tell, introns are going to be in. They're not good. They're going to stay in. But exon are going to exit to be translated. So one of my students asked a very good question. Said, Professor, all these introns, they get to stay behind intron. You talk to us about phenylalanine depositing in the brain. What about all these introns? They get to stick and remain so many inside the nucleus. Isn't that going to damage the nucleus? Is that true or false? Introns. So you know these brown introns, they get to stay in. They're not going out to be with the axons to be translated, right? So if you have a lot of introns piling up and depositing and sedimenting intron, isn't that going to damage the DNA or damage the nucleus? Yes, because it's going to be a lot. So now we have something called alternative splicing, where we recycle, we take all of those little bits of introns and reassemble them to make some useful mRNA from them. This is called alternative splicing. Alternative splicing. Yeah, is when you recycle the left back introns and you make something good out of them. They are turning the lights off. They away, want us out of here. Yes. Post-transcriptional modification, because we already transcribed. Now we gotta modify, do a little surgery, add like a poly A tail to the 3' end, add a 5G cap. Okay, there you go. So you see here, this is a double-stranded nucleic acid. You know that's the DNA, where my mouse is. But this is single-stranded red. You know who's that? That's the RNA. Now when the RNA comes, we need to do modifications. Remember I told you, poly A tail. You add many A tails. That's a post-transcriptional modification. Then you add a 5-guanosine cap. And then you get the exons. And you leave behind the introns. And then you go to be translated in in the ribosome, and then when you translate in the ribosome, you get post-translational modification. We always do modification. You do post-transcriptional modification, and when you do translation, you do post-translational modification. And then you have your fresh, your ready protein. Now imagine a baby is born with a mutation where post-transcriptional modification cannot take place, or post-translational modification cannot take place. Won't that affect the protein product? It will. And if it damages the protein product, it cannot fulfill the function, so the baby is going to be having a disease. Replication, you know, we did the central dogma of biology. Transcribe DNA, you get RNA. Translate RNA, you get protein. But don't forget, you need to sometimes, before you do mitosis, you need to double the DNA, right? Imagine you have a pharmacy, and if you want to make another pharmacy, do you double the medication and take it back to the other pharmacy, or you split your medication in half, one for this pharmacy, one for that pharmacy? You have to double it, because imagine somebody come with a prescription and say, oh, hold on, we split the pharmacy in half. This part, you'll get it over there, but this one, you'll have it here. So it's not going to work that way, because every cell needs all genes. Imagine when you want to do mitosis, you don't have to double the DNA. What if that cell needs hemoglobin? What is it going to do? It doesn't have it because it only has half. So every cell needs a copy of every single gene, because it's going to need every single protein. No cell will never need a protein. So it will all need every single protein. So the same way every pharmacy got to have every single medication, every nucleus have to have every single gene. Okay. All righty. So this is the karyotype. This karyotype is organized from, if you look here, you can tell me, if you look at all these chromosomes, chromosome 1 to 22, these are the autosomal chromosomes, meaning that they are for whole body. But 23, the last one, this is the chromosome for the sex, to tell if you're a male or a female. What do you notice in this karyogram? Well, if you compare 1 to 22, smaller. So they are in the descending order. That's a karyogram or a karyotype. XX means a male or a female. XY means a male or a female. Male. Very good. And you will know why in a little. Because the Y chromosome has a region called the SRY region. If you have the SRY region, you will be a male even if you have 100 X's. If you have a Y region, if you're like that, you're a female. Like that, you're a female, but that is called polyploidy. We're going to talk about it. If you're like this, you're a male. Like this, you're a male. Like this, male. You can have 100 X's as long as there is one Y. You will always be a male. If a Y show up, you're a male. So if I show you a karyotype like that, and I tell you this patient, you never saw the patient. Just look at the karyotype and you saw a little bit of Y. You definitely, absolutely know if you go in the room, that patient look like a man or a woman. Absolutely. Okay? Yes. I mean, if you look at the genitalia, you're going to have, yeah. Are you guys following? Yeah. We will talk about that. But for now, I want you to know that it is the Y chromosome that makes you a male. And the absence of the Y chromosome is what makes you a female, not the presence of an X chromosome. Because we're going to learn about a disease called Turner syndrome. Turner syndrome has only one X, nothing else. Still a female because no Y showed up. One Y show up, you're a male. Why? Because in the Y chromosome, there is a dangerous region, wolf region, called the Wolffian region, called the SRY region. This SRY region is found only on the Y chromosome. If you transcribe the SRY region, you get testosterone. And if you have testosterone, that's it. You will never have a Mullerian duct. You will always have male prototypical genitalia. The SRY. Can we have an SRY region in the X chromosome? In the X chromosome, can you have an SRY region? Impossible. Only for the Y. So we're going to talk about that. So now, autosomes, these are the first 22 of the 23 pairs. Sex chromosome are the 23. Karyotype, this is an ordered display of the chromosomes. So if you look, you need to know what a chromosome is. Those are homologous, meaning they are like mirror images. So for example, in chromosome 1, you have 2 homologous. But you cannot take this one and put it next to this one and say they're homologous. They're not, because those are homologous in 2. Those 2 are homologous. Those 2 are homologous. Those 2 are homologous. But this one and this one are non-homologous. Kinetochore is like a plaque, a protein plaque that the microtubule bind to if we want to pluck off remember anaphase? Remember anaphase? How does anaphase happen? When a microtubule come out the centriole, attach to the plaque, the kinetochore, and separate the 2 chromosomes. What is euploid? This is really, really, really important. Euploid, you always means good. You always means right. It means correct. That's why we don't have, we want eutension. We want euthermia. We want euploidy. We want eucalcemia. We want euphosphatemia. Always you. You means good. You means good. Euploidy, meaning that you have the right good number of chromosome, which is 46. All of us here are euploid. Now, let's look at a problem. Let's say that 2 sperms fertilize 1 egg. Now you have the egg with 1 X, and another sperm with an X, and another sperm with an X. Is that euploid? Is that euploid? Euploid meaning, what are you supposed to have in a zygote? How many chromosomes are you supposed to have in a zygote? 2. Yeah, so one from the, remember they're haploid, diploid. But here you have 2 sperms, so that's triploid. That's no good. That will die. Miscarriage, stillbirth. And sometimes not even the sperm, sometimes the non-disjunction. We're gonna get to that. It's when you have multiple chromosomes, more than 2. So you're supposed to have euploid, haploid plus haploid, diploid. So the euploid is when you're diploid. Now sometimes due to diseases, you have triple X, 4X. Then that is not euploid. That's tetraploid, triploid. We're gonna get to that. Let's just look at it now. So if you look here, haploid and diploid cells are euploid form. So the sperm, 23. That's euploid. Euploid meaning good. Good. Euploid meaning okay. Is the ova, the egg having 23? Is that euploid or not? When the egg has 23 chromosomes, is that euploid or not? Yes, because it's supposed to have 23. The sperm is supposed to have 23. The egg is supposed to have 23. You combine, you get 46. So when the sperm has 23, euploid. When the egg has 23, euploid. When the zygote combine, when both combine, 23 plus 23, you have 46. And that's diploid. Is that euploid or not? Yes, euploid meaning you're good. Polyploid is different. Polyploid meaning you have a multiple of 46. So for example, let's say somebody is supposed to have 46 chromosomes, but they ended up with multiply 46 by 2, 92. What is that person? That is So this is a form of polyploidy multiplied by 3. What is 46 times 3? 46 times 3. You're supposed to have only 46. Euploid is 46. But this person has 2 times euploid. This person has 3 times euploid. That's tetraploid. So polyploidy, meaning your chromosome number is exactly a multiple of your euploid. If you're supposed to be 46. If you're 46 times 2, 92. That's polyploidy. 46 times 3, how much is that? 138. That is polyploid. But let's say somebody has 47. What is euploid? Euploid is 46. But this person, patient number 1, has a 92. 92 is a multiple of 46. So that's an example of polyploidy. But this second patient doesn't have a 92. This patient has 47. Is that polyploidy? No, that's aneuploidy. Aneuploidy. So aneuploidy means the total number of the chromosomes is not a multiple. It could be more or less than the euploid. Euploid is 46. If you're 45, aneuploid. 47, aneuploid. 48, aneuploid. Only when you're exactly at a double or a triple or a quadruple, when you're a multiple of 46, or when you're a multiple of a euploid, that's a polyploid. But when you're not a multiple, you're just any number, it could be less or more. That's an example of aneuploidy. Aneuploid. Aneuploidy is when your chromosome number is less or more than the euploid, but not a multiple. But if you're a multiple, you're a polyploid. Is that clear? Yes. So that means polyploidy is always more. But aneuploidy could be less. You could drop 1. It could be 46, it could be 45, 44, 43, 42, or 47, 48, 49. That's all not a multiple. The multiple are 92, 138. You need to know polyploidy, aneuploidy difference. Is that clear? Yes. Because some diseases are polyploidy, some diseases are aneuploidy. The next thing you need to know, aneuploidy and polyploidy happen in 2 places, in autosomal and germline. Remember earlier I drew for you that we have 2 types of cells. We have 2 types of cells. We have sex cells and we have autosome. Autosome. The sex cells can have aneuploidy and they can have polyploidy. The sex cells can have aneuploidy and polyploidy. The autosomes can have aneuploidy and they can have polyploidy. This is really, really, really important. Polyploidy and aneuploidy can happen anywhere. Remember, we have 2 different types of cells. Autosomes, which is 1 through 22. This one. 1 through 22. Aneuploidy can happen anywhere here. Like, for example, you know Down syndrome? Trisomy 21. So that's in 21. Is 21 sex? Is 21 sex chromosome? Which one is the sex chromosome? 23. So when 21 has an extra chromosome, is that aneuploidy or polyploidy? That's aneuploidy. Is it a sex aneuploidy or is it an autosomal aneuploidy? Is 21. The sex is 23. So when you have an extra in any of these guys, that's an autosomal aneuploidy. When you have a multiple of any of these guys, that's an autosomal polyploidy. But when you have an extra in the sex, that's a sex aneuploidy or a sex polyploidy. So we have 2 types of polyploidies. We will see the diseases now. Some diseases are sex aneuploidy, some diseases are autosomal aneuploidy, some diseases are sex polyploidy, some diseases are sex aneuploidy. Is that clear? All right. And this is what I think she was asking earlier. So if you look here, the problem happens, this is called non-disjunction. We're supposed to have disjunction. Disjunction when we separate. You see, these are two homologous chromosomes. In meiosis 1, the red is supposed to go here and the blue is supposed to go here. Are you following me? This is really, really important. This is the main cell. If we want to do disjunction, blue should have went here and red should have went here. When the disease happened, non-disjunction. Is non-disjunction good or bad? Bad. What is good? Disjunction is good. If the blue would have went here, you would have had blue here and red here. That would have been disjunction, like all of us, thank God. But if both went on the other side and not equal, then that is not, this is non-disjunction. Is that clear? Because in the next round, here you're supposed to have had two reds, and here you're supposed to have had two blue, but there's no blue. So now you're gonna have red, blue, red, blue. So now this sperm is supposed to have one red or one blue. It has one red and one blue. So when it fertilizes the egg already has an X. So this one is coming with two X's. So now there's gonna be three X. That's a sperm with two X's, or maybe a sperm with one X, but an egg with two X's. So now that is going to be an example of aneuploidy or polyploidy. Aneuploidy. Is that clear? So you're going to see here, this sperm is going to have trisomy, it has 3 chromosomes, and this one here has monosomy, it has 1. Are we supposed to have monosomy or trinosomy? Neither. What are we supposed to have? Diosomy. We need both. We need both. Only 2. We only need 2. Okay, now we begin with autosomal aneuploidy. So now from the name, you know that this is an extra or a 1 minus where? In the sex or the autosomes? Autosomes. Now, what is partial trisomy? Partial trisomy, back in the day, they thought the whole chromosome was going to move. No, sometimes a fragment break off and go to the other side. So now you have one whole chromosome and a little bit of shimmy, like a little broken part of another that's a problem. That's a really dangerous problem, because you're supposed to have only one chromosome. Now that is partial trisomy. Partial trisomy is when you have one chromosome with like a extra portion, not a whole thing is present. Still bad. Chromosomal mosaic. What's a mosaic? Mosaic means that you have trisomy, but in like selective parts of the body. Like trisomies are occurring only in some cells, but not in other cells. By the way, what is worst? Polyploidy or aneuploidy? Aneuploidy, because it's better always, both are bad, both are going to have a mental defect and problems in the anatomy of the heart and in the body, big problems. But it's always better to have more chromosomes than no chromosome. It's always, if you had to choose whether you should have less chromosome or more, absolutely more. Absolutely more, having more chromosome. So now when you look at Down syndrome is an autosomal aneuploidy. So you know there's no multiples of the euploid, maybe like one extra. And autosomal, it means that it's in the rest of the body. So one of the most common is trisomy X, meaning that there is you always have 2 chromosomes, right? Homologous. Trisomy meaning one extra showed up. Where? In the 21 region. Where is 21? So this is 21. There is one X, it's called this is Turner. What is trisomy? They didn't even put a karyotype of trisomy 21. This is very bad. So it's supposed to, so here in 21, you will see an extra chromosome here. You see? I cannot believe they didn't even put a. There you go. You see? In 21, you have, everybody has two euploid, except this one has one extra. That's very bad. So now, when you look here, when you look here, okay, that is Down syndrome. Now, this is another disease where female with 3 X's. Turner syndrome is 1 X only. That's why they call it X and O. They call it XO because the sperm fertilized an egg that has no chromosome in it. So it's an X and an O. In an X and an O, is that a boy or a girl? A girl, absolutely girl. Why is it not a boy? There's no Y. If there's no Y, you will absolutely be a girl. That's for Turner syndrome. Yeah, so a sperm fertilizes an egg that has no chromosome, or an egg has a chromosome, but the sperm is empty, has no chromosome in it. So only one has a chromosome. You know, you're supposed to have the sperm has to have a chromosome, the egg has to have a chromosome. But in Turner, one has, the other doesn't. Any combination. So that's why they call it X and O. O in place of the missing one. That's Turner syndrome. Klinefelter is when you have 2 X's and a Y. Now you know what that is. There's 2 X's. That means one sperm, and it's supposed to have only 1 X. It has 2 X's. Actually, the Y only comes from the male. The X is in the female. So the female has an X, and the sperm mistakenly had X and a Y. So this person now will have 3. Is this an autosomal or a sex chromosome? A sex chromosome. So when you look at the karyotype, where's the karyotype for Turner syndrome? They don't have a karyotype for Turner syndrome. So in Turner syndrome, here in 23, you're gonna find 2 X's and one Y. This is absolutely a male, and you know these are the features. Well, you need to know Turner syndrome has webbed neck. Webbed neck. You really need to remember that. Turner syndrome. That is known as webbed neck. You have webbed neck. So whenever you see a patient with a symptom webbed neck, that's Turner syndrome. And this is Klinefelter's disease. Okay. All righty. So we're gonna stop here. And next lecture, before we do the cancer, we're gonna wrap up the. We kind of went over everything actually, but we're just gonna wrap over it real quickly. And please do your homework. Go over all the questions. I published them for you already. Do them, finish them, and go over them as much as you can so you can do well on the exam. Let me take the attendance real quick. Is this last name, first name, or first name, last name?